Mild cognitive impairment is an intermediate state between healthy aging and dementia. 1 out of every 4 older adults suffers from mild cognitive impairment, with symptoms ranging from memory loss and language problems to inability to focus and engage in complex cognition. Every 3rd patient with mild cognitive impairment also has a diagnosis of depression. Having comorbid depression with mild cognitive impairment decreases quality of life and drastically increases the likelihood that someone's mild cognitive impairment will progress into dementia. Combined, these symptoms place a substantial burden on the impacted individuals and their families, healthcare systems, and the entire society. Depression is already the leading cause of disability worldwide, but it is especially hard to treat in older adults. So how do we alleviate depression in older adults and decrease the risk of further cognitive decline leading to dementia? Current treatments are less than optimal, and this could be because they're focused solely on the brain, whereas increasing evidence shows that depression is a whole body disorder. Our multidisciplinary project builds on the idea that depression impacts multiple systems in your body, including your gut. Your gut is home to trillions of microorganisms that synthesize compounds which impact our entire body. This is why the gut is sometimes referred to as the second brain. Our goal is to examine the interaction between mild cognitive impairment, depression, brain connectivity, and the microbiome. This is the essential first step towards developing novel, non-invasive treatments that will be efficient in late-life depression. How are we going to do this? First, we use cognitive and behavioral assessments, for example, questionnaires and interviews. This allows us to establish what symptoms participants are experiencing. Next, we know from previous research that depression is associated with robust signatures of disrupted brain network communication. Here, we use functional magnetic resonance imaging to look at brain activity and establish how the brain changes with depression. Additionally, we use stool samples to perform high-resolution gene sequencing. This allows us to determine what bacteria live in the guts of our participants. Maybe those with depression will have higher levels of bad, pro-inflammatory bacteria in their guts. Finally, we will use computational modeling to build the first model of the gut-brain behavior function in older adults with mild cognitive impairment and varying symptoms of depression. The number of elderly in the world is predicted to double by 2050, and with the prevalence of late-life depression and dementia drastically increasing, we need to act now. This project is the critical first step towards novel, non-invasive treatments that will effectively alleviate symptoms of late-life depression and decrease rates of dementia.